Okay, this is a verbal, no video. So you can listen to this while you're chilling, while you're going to sleep. The reason I'm doing this is because I'm going through the same class you are, Microbiology 1, whether you're doing this in university, community college, or a city college. You're all going to be reading the same book. And I was just so tired because I'm taking anatomy and physiology too, and microbiology and astronomy and algebra right now. And I'm too old for this shit. So one thing I can tell you is reading the book is the hardest thing. And you have to read the book to pass these classes. If you read the book, everything's in there. It's all the same. Okay. So this book is by Wiley. Black is the author, I think. Okay. Good book. All right. So there's an area here with a bunch of vats, and it says, when you sip wine or cheese, do you ever wonder about the microorganisms necessary for making those products? Where do they come from? How are they stored? They might be kept at ATCC, American Type Culture Collection, in Manassas, Virginia. ATCC is a global nonprofit organization that preserves, authenticates, and distributes microorganisms and other biological materials for the scientific community. In addition to ensuring that high-quality microbial cultures are available for research and development, ATCC serves as a safe deposit location for industrial cultures owned by commercial organizations, including winemakers, brewers, cheesemakers. ATCC stores these valuable cultures under multi-layered security system in large liquid nitrogen tanks at very low temperatures. Manufacturers of wine, cheese, or other products requiring microbes can safely preserve their cultures at ATCC until they need to replenish their stocks. Other cultures stored at ATCC are available for use in research and development projects in many fields. ATCC scientists ensure that cultures used by researchers are correctly identified and the stains, genetics, genetic and biochemical properties are maintained. Okay. Now, it talks about, we're on chapter 9, talks about taxonomy, the science of classification. Linnaeus, 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 the father of taxonomy, using a taxonomic key. Problems in taxonomy, development since Linnaeus' time. The 5-kingdom classification system. You've got Kingdom Monera, same thing. Kingdom Protista, Kingdom Fungi, Kingdom Plantae, Kingdom Animalia. Now, if you've taken biology too, there are some differences there. I'm not even going to get into them now. I've taken that class. You have to really be into science to want to take that class. The 3-domain classification system. The evolution of prokaryotic organisms. The tree of life is replaced by a shrub, the archaea. That sounds really stupid. I don't even understand that myself. I guess they're just talking about prokaryotic organisms and the archaea. That's it. Now, classification of viruses, the search for evolutionary relationships, special methods needed for prokaryotes, numerical taxonomy, genetic homology, Other techniques, the significance of findings, bacterial taxonomy and nomenclature, criteria for classifying bacteria, the history and significance of Berge's manual, problems associated with bacterial taxonomy, bacterial nomenclature, bacteria, taxonomy, and you. Okay, I'm going to take a little sip here. There's a lot of reading to do. And this is me doing the reading. I would never read this again, probably. I would go down to the slides after this, but you have to always read. Humans appear to have an innate need to name things. In many primitive societies, a person who knows the true name of an object or of another person is believed to have power over that person or object. Naming helps us to understand our world and to communicate with others about it. Now, there's a picture on the page of Carolus Linnaeus. I got this book used, and somebody put asshole next to his picture. I guess they didn't like naming things. Anyways, now, what was I saying? Okay, so we're talking about naming things. Now, taxonomy is the science of classification. You know they're going to ask you that question. In science, accurate and standardized names are essential. All chemists must meet the same thing when they talk about an element or compound. Physicists must agree on terms when they discuss matter or energy. And biologists must agree on the names of organisms, be they tigers or bacteria. Faced with the great number and diversity of organisms, biologists use the characteristics of different organisms to describe specific forms of life and to identify new ones. The group of related organisms together is the basis of classification. The most obvious reasons for classification are: number one, to establish the criteria for identifying organisms. Number two, to arrange related organisms into groups. And number three, to provide important information on how organisms evolved. Now, you know they're going to ask that question, so I'm going to read it again. So, the grouping of related organisms together is the basis of classification. So they're probably going to say, what is classification? Or name the three things having to do with classification. So you have the most obvious reasons for classification are, number one, to establish the criteria for identifying organisms. Number two, to arrange related organisms into groups. Number three, to provide important information on how organisms evolved. Taxonomy is the science of classification. It provides an orderly basis for the naming of organisms and for placing organisms into a category, or taxon. And then it says, in parentheses, plural, taxa. Now, the best thing to do is, if you're tired of hearing this, just pause it and listen again and again and again. I listen to videos constantly, but I didn't see any good ones on microbiology. So I hope this helps. Anyways, you can make fun of me too if you want to, but I'm just reading from the book. Another important aspect of taxonomy is that it makes use of and makes sense of the fundamental concepts of unity and diversity among living things. Organisms classified in any particular group have certain common characteristics. That is, they have unity with respect to these characteristics. For example, humans walk upright and have a well-developed brain. Escherichia coli, E. coli, okay? I don't even know if I pronounced that correctly. Cells are rod-shaped and have gram-negative cell walls. You're going to be working with E. coli. You probably have done, you had to have done it already, the E. coli. Just disgusting, but it's in your duty. And gram-negative cell wall. The organisms within taxonomic groups exhibit diversity as well. Even members of the same species display variations in size, shape, and other characteristics. Humans vary in height, weight, hair, and eye color, and facial features. Certain kinds of bacteria vary somewhat in shape and in their ability to form specific structures, such as endospores. A basic principle of taxonomy is that members of higher level groups share fewer characteristics than those in lower level groups. Again, a basic principle of taxonomy is that members of higher level groups, so that would be like humans, us, and Homo sapiens, share fewer characteristics than those in lower level groups. So the lower you go, the less characteristics you share. Like all other vertebrates, humans have backbones, but humans share fewer characteristics with birds, fish, than with other mammals. Likewise, nearly all bacteria have a cell wall, but in some, the wall is gram-positive, and other, it is gram-negative. Remember, gram-positive, giant peptidoglycan, gram-negative, peptidoglycan in the center, like an ice cream sandwich, and acid-fast, just a thick lipid layer. And then Linnaeus, the father of taxonomy. The 18th century Swedish botanist Carlos Linnaeus is credited with founding the science of taxonomy. See figure 9.1. He originated binomial nomenclature. Binomial nomenclature. The system that is still used today to name all living things in the binomial or two-name system. The first name designates the genus, or plural is genera, of an organism, and its first letter is always capitalized. The second name is the specific epithet, and it is not capitalized. Epithet, E-P-I-T-H-E-T. And it is not capitalized, even when derived from the name of the person who discovered it. Together, the genus and specific epithet that identify the species is genus species, to which the organism belongs. Both words are italicized if done on the computer, or if they're done in print, then you just underline if they're handwritten. So both words are italicized in print, but underlined when handwritten. When there is no danger of confusion, the genus name may be abbreviated to a single letter. Thus, Escherichia coli is often written E. coli, and humans, Homo sapiens, may be identified as H, capital H, period, and small S-A-P-I-E-N-S. The name of an organism often tells something about it, such as its shape, where it's found, and what nutrients it uses, who discovered it, or what disease it causes. Some examples of names and their meanings are shown on Table 9.1. The members of a species generally have several common characteristics that distinguish that species from all other species. As a rule, members of the species cannot be divided into significantly different groups on the basis of a particular characteristic, but there are exceptions to this rule. Sometimes members of a species are divided on the basis of a small but permanent genetic difference, such as need for a particular nutrient, resistance to a certain antibiotic, or the presence of a particular antigen. When organisms in one pure culture of a species differ from the organisms in another pure culture of the same species, the organism in each culture are designated as strains. A strain is a subgroup of species with one or more characteristics that distinguish it from other subgroups of that same species. Each strain, identified by a name, number, or letter that follows the special epithet, for example, capital E period, small coli, strain K12 has been extensively studied because of its plasmids and other genetic characteristics. And E. coli strain 0157, capital H7, causes hemorrhagic inflammation of the colon in humans. I hate when that happens. In addition to introducing the binomial system of nomenclature, Linnaeus also established a hierarchy of taxonomic ranks: species, genus, family, order, class, phylum, division, and kingdom. You should just know that like you know your last name. I mean, just be able to whip that out. Kingdom, division, phylum, order, class, family, genus, species. At the highest level, Linnaeus divided all living things into two kingdoms, plant and animal. And I hate when they do that, tell us about something in the past that confuses us about something that it's actually supposed to be. So I'm going to read this again. In addition to introducing the binomial system of nomenclature, Linnaeus also established a hierarchy of taxonomic ranks: species, genus, family, order, class, phylum, or division, and kingdom. At the highest level, Linnaeus divided all things into two kingdoms, plant and animal. In his taxonomic hierarchy, much of which is still used today, each organism is assigned a species name, and the species of very similar organisms are grouped into a genus. As we proceeded up the hierarchy, several similar genera are grouped to form a family, several families to form an order, and so on to the top of the hierarchy. Some hierarchies today have additional levels, such as subphyla. Also, it has become accepted practice to refer to the first categories within the animal kingdom as phyla, and to those within other kingdoms, we now have 5, as divisions. Recently, the 5 kingdoms have been grouped together into 3 domains, a new category even higher than kingdom. Domains will be discussed later in the chapter. The classifications of a human, a dog, a wolf, and a bacterium are shown in figure 9.2. Using taxonomic key. Biologists often use a taxonomic key to identify organisms according to their characteristics. The most common kind is a dichotomous key, which has paired statements describing characteristics of organisms. Paired statements present an either or choice, so that only one statement is true. Each statement is followed by directions to go to another pair of statements until the name of the organism finally appears. There's a chart. Is a dichotomous key that will identify each of the 4 most common U.S. coins, quarters, dimes, nickels, and pennies, read statements 1A and 1B, and read statements 1A, 1B, and decide which statement applies to a given coin. Look at the number to the right of the statement. It tells you which pair of statements to look at next. Continue in this manner until you reach a group designation. If you have followed the key correctly, that designation will name the coin. Of course, you don't need a taxonomic key to identify something as simple and as familiar as coins, but identifying all the many kinds of bacteria in the world is a more difficult task. Major groups of bacteria can be identified with the key in 9.4. More detailed keys using staining reactions, metabolic reactions, fermentation of particular sugars or release of different gases, growth or different temperatures, properties of colonies or solid media, and similar characteristics of cultures. By proceeding step by step through the key, one should be able to identify an unknown organism or even a strain if the key is sufficiently detailed. So a good thing would be to pause, and if you have the book, look at the key. Problems in taxonomy. So that's why people should get the book somehow. It's not my, they're not giving me money to sell books. When they do, I'll start telling everybody about exactly what the book I'm using and how wonderful it is. Problems in taxonomy. Among the aims of a taxonomic system are organizing knowledge about living things and establishing standard names for organisms so that we can communicate about them. Ideally, we would like to classify organisms according to their phylogenetic or evolutionary relationships, but this is not always easy. Phylogenetic is spelled P-H-Y-L-O-G-E-N-E-T-I-C. Phylogenetic or evolutionary relationships, but this is not always easy. Evolution occurs continuously and at a relatively rapid rate in microorganisms, and our knowledge of the evolutionary history of organisms is incomplete. Taxonomy must change with evolutionary changes and